following is a conversation with Dr. Momo Vuyisich, co-founder and chief scientific officer at Viome. Viome is a life sciences company that analyzes the activities in your oral microbiome, gut microbiome, and blood using RNA, essentially measuring gene expression. This gives the company the ability to be able to assess what the microbiome in these various different areas of the body is doing, and gives insights into potentially how that links to diseases, and gives you personalised recommendations for how to improve your microbiomes through your environment. So basically your diet. Their vision is that in the future, through the analysis of all of this data, they're able to make predictive bets on who is at higher risk of developing particular diseases and issues due to the composition and function of the microbiome. They're able to develop earlier diagnostics based on these data, and ultimately also develop therapeutics. Momo starts the pitch within the podcast with a very, very grand and big vision about how in the future he believes that all chronic disease, including cancers, Alzheimer's, Parkinson's, things that we'll all be familiar with and that will have touched us at some point in our lives, either directly or indirectly through family and friends, will all be thought of as like the bubonic plague. Insofar as at the time when the plague was a real issue, people were dying and people didn't understand anything about it. Why was this happening? And his view is that through the applications of technologies and platforms like the one they've developed at Viome, these diseases that we speak of will ultimately become diseases of the past in the history books. Because ultimately we are just big bags of chemical reactions, and we just don't understand the science well enough yet to be able to understand why these things develop. And it is, of course, immensely complicated. And it takes time and big data and platforms and crunching and human knowledge and machine knowledge to be able to untangle and decipher all of these things. And we have made so much fundamental progress as a human race, so to speak, over the centuries and decades. We now understand the biological causes and basis and pathogenesis of many different diseases, but we're not there yet. When President Bill Clinton and Prime Minister Tony Blair stood up and announced to the world just after the dawn of the new millennium that society had achieved its greatest ever scientific feat, its greatest milestone, we had sequenced the human genome as a result of the International Human Genome Project. What they said in their pitch were some big, big statements around how in the coming decades we'll view cancer as just a constellation of stars. Well, here we are in the year 2024, and that's over 2 decades. And actually, we probably know less about cancer than we thought we did then, insofar as it's not a cracked science. It's almost like the more we figure out and the more we understand, the more we realize we don't know, which is the same in many different things. And we are nowhere near cracking cancer. We are a long way away. What we have developed are some amazing therapeutic approaches. In particular, and we've spoken about it on this podcast a couple of times, immunotherapies, so ones that modulate the immune system. And now, because we're understanding our microbiome, there may be ways to improve the efficacy, so how these treatments work in patients by changing the microbiome and modulating the immune system. That's really, really exciting. Now, Viome is an at-home collection platform company. That means that you can send your samples off to Viome, they analyze them, and they send them back to you. And what you can expect to learn in this episode with Momo is how Viome came to be and how they've built the company from a spin-out from his academic lab into a world leader in microbiome analysis. That story is really, really interesting. Momo is an amazing communicator. You'll learn about the microbiome and its importance in gut health. You'll hear about his own story in terms of how he managed to cure a chronic disease through his own diet. You'll hear about his vision for Viome, how the pharma industry works in his opinion, and how things are going to improve over the coming decades. Fascinating discussion. Really, really enjoyed it and wanted to thank Momo for coming on as such a busy person. With the introduction done, I wanted to say thank you to all of our listeners for supporting the podcast. Some of you are listening to every single episode and sending feedback about the episode, what you liked, what you didn't like, what you learned. That's awesome. It makes it all worthwhile. We've been going now for, a year and a half, and we're loving every single moment. And the feedback from listeners makes it all worthwhile. So if you're enjoying it and you haven't yet liked and subscribed, please hover your finger over that button and give us a 5-star review. It really helps keep the podcast going. So without further ado, I will now hand over now to the episode. This is Inside Matters. My name is Dr. James McElroy. Thank you for taking an interest in your own gut health and mastering your microbiome. So let's get right into the thick of it then. Can you articulate your vision for the future and how it pertains to microbiome science and chronic disease?
Whoa, yes, diving right in. So, so the future I am working hard on building, and I would like everyone else to be building, is a healthy future where chronic diseases and cancers are understood just as well as we understand acute infectious diseases. And so let me throw in a metaphor that in the Dark Ages, we had the Black Plague, and it killed a third of the world's population. And at that time, it was just bad luck. If you get it and die, it was literally bad luck. And that's because we didn't understand what caused it. We didn't understand how it was transmitted. We didn't know how to prevent it. We knew nothing about it. It was basically you either live or die, and it's bad luck if you die. And today, 700 years later, all chronic diseases and all cancers are in the exact same bucket. If you get multiple sclerosis, oops, bad luck. And you're going to die a slow, miserable life. If you get ALS, bad luck. You're going to die a slow, miserable life. Cancer, you're going to have a very dramatic life and you may or may not die, but it's all a matter of luck. However, none of the chronic diseases and none of the cancers are bad luck. They're simply biochemistry that we do not yet understand. That's it. And so that's one point to make, that these diseases and cancers are just human physiology that we do not yet understand. And second point, which I think is even more important, is that every single piece of science and technology needed to understand the root causes of every chronic disease and cancer and to prevent them exists today. All that we have to do is do science differently from the way it's being done today. And we will absolutely be able to understand the root causes and how to prevent them. And that's really one of the highlights and big takeaways from this podcast that I would like everyone to understand.
It is a big vision. So the next question is, how do we get there? What are the tools, the workflow?
So I think this concept of we have to do science differently is the key. And let me just explain some things. So I'll go at a high level and then we can dive deeper as your interests guide us. So we think as members of the society, or we grow up thinking that we have lots and lots of science that's improving human health. But then when we really break it down and we look at the incentives, the academia are incentivized by publications. Their salaries, their promotions never depend on, did they change humanity? Did they improve humanity? Did they improve human lives? That doesn't factor into their promotions and salary increases. It's only papers. And you can publish papers without discovering anything relevant to human health or anything relevant to improving humanity. In fact, the vast majority of papers are that, Even if you go and, let's say, do this exercise, go to Nature, Cell, and Science publications from the year 2000, let's say, and pick 100 random ones. And even the covers, pick the covers, the best ones. What is a typical outcome of reading? You're going to find out that they discovered something, and at the end of the abstract and the end of discussion, it'll say, this could lead to, and then some grandiose goal of better cancer care or, better improved something. Well, 24 years later, ask yourself, did that really happen? And the answer is, it hasn't. And so this is a standard template for academic publications. We discovered something, it could be important, and it could lead to something, but it doesn't, because we don't translate that knowledge into something that's useful to humanity. So that's one absolutely major problem with academia. key theme is academia are driven by publications, not by improving humanity. And if we can just change that incentive, we have super smart academics. We have very well funded, very well equipped, very well resourced with smart scientists. We don't need to change anything about the scientific enterprise. We just need to change the incentive. And we can utilize the same human, material, and financial resources to completely change the world in a very, very profound way. So that's academia. And I'll discuss in a brief moment how I think we can change that, because I've written a paper on that with a team of mine. The second is pharma. People still today, many people, or maybe most people, think that pharma is there to actually make you healthier. But that is not the case. Pharma's business model is not to cure diseases. It's not to prevent diseases. It's not to make you healthier. It's to manage your illness. Their business model is to make money off of continuously for the rest of your life, managing your illness and drawing financial gains from that. That is the business model. And that doesn't mean that people in pharma are evil. That doesn't mean that the VCs who are funding pharma are evil. It's just a business model. That platform is meant to provide drugs that manage your illness and draw as much money out of it as possible. That's it. And that's how it is. And so when I had an autoimmune disease that was, going to head me into a wheelchair, I had I did an analysis of these large human, societal systems that were supposed to help me, and they were not in fact helping me. And so I realized that we need to create an entity that is actually focused on identifying root causes of chronic diseases and cancers, and then finding ways to prevent them first. And then if they do happen, how do we reverse them? And so that's why we created that. That's why I developed a technology at my national lab in Los Alamos for 6 years, and then spun out the company Viome. And now we've been working on that at Viome for almost 8 years. So it's been a 14-year journey, basically. Okay, so now let's talk about the real question. How do we actually direct this scientific enterprise that has hundreds of thousands of ultra smart people, ultra hardworking people, very well equipped laboratories, lots and lots of money is poured into this? How do we just change the incentive model and go for changing the humanity, improving humanity? So we don't have to reinvent the wheel. This has already been done. To me, there are several examples of that. And I'll specify those. And so the Manhattan Project, I think, is the first one, and I still think it's the best one.
Yep.
But then we can also look at the Apollo programs, where America sent people to the moon. We can also even look at Tesla as an example of such a focused translational science. And there are many other such programs, but I really want to hone in on the Manhattan Project. And, before Oppenheimer movie, I was sort of hesitant to bring the Manhattan Project because I first had to explain to people what it was. But luckily now, since Oppenheimer movie, people are like, oh yeah, I know the Manhattan Project. But let me explain the Manhattan Project that is exciting to me. Let's forget about the purpose of the Manhattan Project in terms of building a nuclear weapon. And let's not get into that discussion of nuclear weapons. Let's talk about the operational excellence and mission of such a project. So the US government said, we want to do something that has never been done before, that the vast majority of people thought it was science fiction, right? And the US government said, let's gather the smartest people, let's put them in one place so that they can work together every single day. Let's give them unlimited resources, human, financial, material, all resources. Let's remove the burden of any bureaucracy. That's a big deal, bureaucracy.
That's a very big deal.
That's a big deal. And let's give them one incentive and one mission only. None of those people were asked to publish any papers. None of those people were asked to build universities. None of those were asked to bring PhD students to life. None of them were asked to do anything. Every single person, there were 100,000 people on the Manhattan Project. Every person, every single day had one thing in mind: to build the atomic bomb. And if we have such a project, a Manhattan Project for chronic diseases and cancers, where we say we are going to identify root causes of multiple sclerosis. Forget about the papers, forget about the PhD students, theses, forget about IRBs, forget about all the bureaucracy surrounding clinical research. Let's bring the world's leading scientists, physicians, technologists, and put them together who have been studying multiple sclerosis and give them one goal, give them unlimited sources of funding, unlimited number of people, unlimited facilities, and their goal is in a few years, 2 to 5 years, to identify all root causes of multiple sclerosis and then provide possible solutions as to how we can prevent them with nutrition, with lifestyle, potentially pharmaceuticals, potentially existing or new pharmaceuticals, things like that. And then let companies basically feed on that information and come up with commercial solutions.
With solutions.
Where, for example,
that's interesting.
Yeah. Where, for example, if the Manhattan Project for Multiple Sclerosis says that if you are colonized by these 3 species of fungi that produce these mycotoxins, and you have a leaky gut because you do not have these 3 microorganisms in your gut that allows the colonization and that allows the translocation of mycotoxins into the bloodstream, and you have too many of these 3 microorganisms that are causing LPS production and blood-brain barrier permeability that are allowing these mycotoxins to go into the central nervous system. Now you get multiple sclerosis. And now companies like yours, mine, many others will say, oh, well, so we need to boost these 3 microbes. We need to reduce these 3 microbes. We need to have a test for these 3 fungi. We need to have a test for leaky gut and permeability of blood-brain barrier. And now we can truly understand and profile people and tell every single person every 6 months, you will not get multiple sclerosis. You just don't have the factors that are needed to develop multiple sclerosis. Or you do a test on a person and you say, wow, of the 4 factors needed to develop multiple sclerosis, you have 2. We may want to tweak your gut microbiome. We may want to tweak your diet in order to reverse those 2 because it's a slippery slope. And that would be basically the Manhattan Project for multiple sclerosis. And then we need to have a Manhattan Project for Alzheimer's and Parkinson's and type 2 diabetes and every type of cancer, so that we can then profile humans every 6 months and show them a dashboard, literally with a gauge for every chronic disease and cancer. And that gauge says, oh, you're in the green for multiple sclerosis, or you're in the orange, do this, or you're in the red, you're in danger zone. For every disease, you would have a gauge. Just like you walk into a cockpit of an airliner and you see a gauge for every single system on that airplane, why can't we bring that to humans for their physiological systems?
If I just make a comment, I think some may say that the Human Genome Project was maybe an example of international collaboration to deliver a specific goal, which was to sequence a human genome. And if you listen to Bill Clinton at the dawn of the new millennium, he does say, within the next couple of decades, we'll view cancer as just a constellation of stars, which obviously, as we sit here in 2024, is not how we view cancer. So how does the microbiome play into all of this, in your opinion, given that we're on a gut health microbiome podcast?
Well,
into the big vision.
You actually said a lot in a very few words, which, that was pretty amazing. So let me comment first that the Human Genome Project was, in fact, one of these large, highly focused projects where publications were not the main thing. It was sequence the human genome. So that's another example of such focused projects in many institutions. I think the Manhattan Project is still better operationally than the Human Genome Project, but that's a similar concept. So love that. Second thing about the human genome is back in the 90s, when this proposal came out and this project came out, we didn't know about the gut microbiome. We didn't know about the importance of it and that we were a holobiont. We were actually not human. We look like human, but we're in fact mostly microbial. We just simply didn't know that. And I think that it was a very natural thing for humans to say, hey, the genome is inherited. This is what makes me who I am. Let's sequence it. And I think whether the promise of, this is going to unravel all of the disease mysteries of the human was honestly thought to be the case, or it was just a sales strategy, at this point in time, it's a moot point, because the Human Genome Project was very important. And human genetics does play a role in chronic disease and cancers to a small degree. This is actually, I think, one of the most important parts of this discussion. The biggest takeaways from this conversation, I think, that people should have about the human physiology is, think of the human body as a synergy between our own genes and microbial genes. And so think of us as a vessel with hundreds of thousands of chemical reactions that are driven to a large extent by the nutrients that we obtain from our food. And so that's a very important aspect, to think that we are basically 100% built from nutritional ingredients that we consumed. I mean, when you're born, you're basically 3D printed from the foods that your mom ate. You're 100% made up of those ingredients. How crazy is that, right? And so how do we funnel? It is crazy. So how do we funnel those nutritional ingredients into 3D printing our body? They affect directly our own genes, but they affect directly a lot of microbial genes that then affect our own genes. And that's the evolution. We have co-evolved with these microbes. There are many plants that have more genes than humans, and yet those plants don't have fancy immune systems, fancy brain, fancy organs like we do, right? But they have more genes because they need to depend more on their own genes, whereas we have evolved with these microbes, and so we don't have to have a lot of our genes. Unfortunately, these microbes can be transient. They're not basically inherited, which means that while we used to live with nature and we used to basically propagate these microbes from person to person, from generation to generation, now with modern lifestyle, we are starting to lose a lot of them because we're living too healthy of a lifestyle, too clean of a lifestyle, not healthy, sorry, too clean of a lifestyle, and we use too many antibiotics and so on. So how does microbiome contribute to our overall physiology? The microbiome, first of all, feeds on the micronutrients that we consume. And during the process of feeding, it expels or secretes other, what we call metabolites, but you can call them molecules or chemicals or many names, but they're basically molecules that we call metabolites. So I'm going to give the simplest possible example of such a function, a microbial function. You're a Scot. I assume you like beer.
Oh, yeah.
So, exactly. So making of the beer is exactly what happens conceptually in our gut microbiome. You provide a substrate, which is barley, and you provide a microbe, which is the yeast, the brewer's yeast, Saccharomyces cerevisiae. That yeast expresses the genes that enable it to chop down the polysaccharide in barley, import the individual pieces of that polysaccharide, which is now glucose basically, and then ferment that glucose into ethanol. And during that process of converting barley into ethanol, and ethanol is that byproduct or that metabolite, during that process, the yeast harvests the energy, the chemical energy from glucose into ethanol, and that's how it lives. That's how it builds itself and divides itself. So it is feeding on barley and lives and propagates, and the byproduct of that feeding is ethanol, and that's what we like. Mm-hmm. And so as a metaphor, I call that micropoop from the microbiome. So ethanol is basically micropoop. The yeast literally poop it out because they don't have anything, they can't do anything with it, and yet we enjoy it.
We love it.
That's a new concept. Now when you go to a pub tonight, you can think about that, micropoop. So we, in a typical human, will have something on the order of 1,200 species, as far as we know today, 1,200 different species of microbes, and they all are there because they've been feeding on the food that you have been ingesting, and they all feed on different micronutrients that you're introducing, and they're all producing these byproducts. So whenever they feed on something, it doesn't just disappear. It actually always gets converted into some byproduct that we call metabolite. And this soup of metabolites turns out to be super important, and we have co-evolved to depend on these metabolites to run our own physiology. And we can have numerous examples, but I'm going to give just a couple. Is that okay? Of these metabolites?
Please.
So one of the most important groups of metabolites that the gut microbiome produces are called short-chain fatty acids. So these are acetate, propionate, and butyrate. And if we just look at butyrate, this is like a magical molecule that if we do not have butyrate, here are the consequences of that. So we do not consume butyrate, and our genes are incapable of making butyrate. Butyrate is basically made specifically by gut microbiome in the colon in response to fibers or prebiotics that we consume, vegetables. And so this butyrate does several very important things. First of all, it actually feeds our intestines. So our intestinal cells use butyrate as food. So the rest of our body uses typically glucose as food, but intestinal lining cells have evolved to use butyrate as carbon source. Second thing that butyrate does is it regulates gene expression of our own genes. So it directly affects our physiology. And one of the most, I think, dramatic examples of that is the conversion of cytotoxic T cells to memory T cells. So I love this example. So we all know people who get sick all the time and even repeatedly get sick with COVID or the flu. And then we know people who either get vaccinated or get that disease once and they're done. All right. So how does this work? We have an immune system that has the ability to develop a memory to past infections. And so if you have COVID or you are vaccinated with COVID vaccine, there is a part of the immune system called T cells, and these T cells attack our own cells that are infected by the virus to kill them, and they clear the virus and we get healthy. But at the tail end of that infection, those T cells will either die or they will get converted into memory T cells. The signal that is required for their transformation into memory T cells is butyrate that is produced by the gut microbiome. So without that signal, the T cells that just fought off this infection die off. And guess what? Next time you get COVID, your immune system won't remember and you'll get sick again. But if you are at that time are eating the right prebiotics, have the right microbiome members that will convert it into butyrate, and butyrate translocates into the bloodstream, it will signal T cells to convert to memory T cells, which will survive that infection and stay in your bloodstream for decades. And next time you get COVID, those cells are already there. They recognize COVID. They start dividing, and you clear the infection before you even develop symptoms.
So should we be giving our vaccines with a probiotic and a prebiotic?
I think so. Yeah. Well, probiotic for some people, unfortunately, we have very few butyrate producers as probiotics, right? We now have Clostridium butyricum that's available, and Akkermansia can also make butyrate.
Yeah, I've seen that.
Faecalibacterium prausnitzii is one of the most well-known butyrate producers, and I don't think it's available yet as a probiotic, but I do think that companies are working on it.
That's right. That's my understanding as well. It's not commercially available, but it's been developed as a drug, so that could be the future.
So yeah, I think one day that's exactly what's going to happen. Go ahead.
Yeah. If we could just bring you back, then you were going to give another example, and then I'll take you to another part of the conversation where we talk about how the microbiome has changed over the decades and whether it's a kind of cause or consequence of disease process. But before we go there, you were going to talk about another molecule in addition to short-chain fatty acids.
Yeah, let's talk about another molecule. So let's make this quick because people can read about this. It's just now everywhere on the internet. So the molecule that I want to point out is these are molecules known as lecithins, and they are basically fatty molecules that have choline in them. And if you have the right microbiome, and if those microbes are not busy consuming other foods that you consume that are more energetically favorable for them, they will consume choline and they will convert it into something called TMA or trimethylamine. And that trimethylamine is a metabolite. It's, again, a byproduct of their own metabolism where they harvest energy from our own food. And that trimethylamine will translocate into the bloodstream, and our liver will oxidize it into trimethylamine oxide, TMAO. And this TMAO molecule turns out to be very dangerous for cardiovascular disease. And so I just encourage readers who are interested in this topic to go look at TMAO and how profoundly is connected with the onset of cardiovascular disease. And I can mention one more molecule. It's phenylacetylglutamine, which is basically a 3 amino acid peptide that's derived from protein fermentation in the colon. And this is now a newer molecule that's discovered since TMAO was discovered over a decade ago. And phenylacetylglutamine, I also encourage people to read about because there's some really strong causative, not just associative, but causative evidence that it causes cardiovascular disease. And again, it's a byproduct of fermentation. And so to me, that would be a warning sign that if you eat too much protein and a lot of protein makes it into the colon, there's a danger that your bacteria will convert it into phenylacetylglutamine, which will cause potentially cardiovascular disease.
Thank you. And that segues perfectly into what I was going to ask you about next, which is this cause or consequence discussion, chicken or the egg. And what can you say to the listeners about our understanding of the microbiome's importance in disease processes?
Yeah, I think the depletion of the gut microbiome is one of the major root causes of the chronic disease epidemic that we have and the cancer epidemic that we have. And that's sort of a very long discussion, but I'll summarize some major points. As we discussed earlier, the functions of the microbiome are critical for our health. They're absolutely critical, but there are functions of the microbiome that also cause disease. And so a couple of examples that come to mind are there's a molecule type called lipopolysaccharide or LPS, and people can read about the dangers of LPS production by the gut microbiome. But recently, just like a couple of weeks ago, someone published a preprint showing that they identified very strong epidemiological evidence that low-level that vaccination against Clostridium tetani is strongly associated with a reduced risk of Parkinson's disease. And they argue that Parkinson's disease could be caused either partially or wholly by colonization of Clostridium tetani. So it's not an infection where you die, but it's a small, low-level colonization where that Clostridium tetani produces small amounts of the tetanus toxin, which then causes the death of neurons. And so, that's really a big deal for the Parkinson's community. But there are many such examples where having the right microbiome mixed with the right diet that produces the right metabolites that support your healthy physiology, and not the wrong microbiome or the right microbiome mixing with the wrong diet to produce unhealthy metabolites that are toxic to your body. So it's really that balance. And so the reduction of functions in the microbiome, I think, is the major cause of chronic diseases and cancers. And let me give you one really, really nice causative example. So if you I'll give you actually two because they're both very dramatic. There are now 2 peer-reviewed publications that describe bipolar patients who were resistant to pharmaceutical interventions. And they had basically a devastating bipolar disorder that basically did not allow them to live a normal life, social or professional. In both cases, a repeated fecal microbiota transplants completely cured them of the disease to where they got off the drugs and they displayed no symptoms, right? So if you ask 100 psychologists and psychiatrists, what is the bipolar disease of? They would say, well, it's a brain chemistry, right? Because that's what they've been taught. But it may turn out to be not the case at all, that it's actually the gut chemistry where these people have lost very, very important members of their gut microbiome that are producing chemicals that are traveling up the vagus nerve, or they're signaling the vagus nerve to cause brain changes that we call bipolar disorder. But if you replenish those gut microbiome members, boom, the disease is gone. You're balanced and there was nothing wrong with your brain. It was actually the symptom of the lack of those members. So that to me is a very dramatic example because it's causative, right? You did something and it changed this person's life completely. Second example,
was this a trial or was it a case report? Sorry, Momo. Was it a trial or
case report? Yeah, two individual case reports, N of 1 each. The next example that I want to give is also very dramatic. So two institutions, very highly regarded institutions, the NIH in the US and the MD Anderson Cancer Center in Houston, Texas, they both repeated this approach. They basically took melanoma patients. Melanoma is a very dangerous type of cancer. They took melanoma patients and treated them with immunotherapy. And they had patients who responded to immunotherapy and basically cleared the cancer. So those are responders. And then they had patients who did not respond to immunotherapy, the same immunotherapy, and they were non-responders. Well, we could say, bad luck, sorry, you're going to die. But they made the extra step and hypothesized that gut microbiome played a major role in this response. So they actually took poop from responders, gave it to non-responders, and the non-responders became responders. So now this is really science fiction before this was published, this would have been science fiction, but now it's real, where you have the same person, same genetics. They have the same cancer that they had a month ago. They eat the same food. They live in the same environment. Everything is the same. All you did was install some extra members of the gut microbiome from the responders, and you converted their cancer into a responder. I mean, that is an incredible finding to me in terms of demonstrating the power of the microbiome.
Yeah, it's a beautiful example because it is the classical assessment of causality, really, isn't it? You know, you actually move the microbiota from someone who has responded into the microbiome of someone who hasn't responded, and you flip their outcome. Those publications were, I think, a couple of years ago now in Cell, and we still haven't seen very significant investment into companies pursuing that approach. You speak to some people who are in the larger pharmaceutical industry, and based on my experience, they don't actually believe the results. They say, yeah, a small study needs to be replicated, and we're still not quite sure the microbiome is important. And I struggle to get my head around that, to be honest.
Yeah, well, I think with absolutely anything in life, it doesn't have to be science or medicine or health or anything like absolutely anything else in life. Nothing is perfect, meaning that if your goal is to discredit something, you can always find holes in anything, no matter how extraordinary it is, right? The best events in the world can always, if your goal is, oh, well, but this was not good, or something bad happened here. I think it's a revolutionary approach, but I think it can be improved significantly. And the reason I think, so as a scientist and a microbiome expert and so on, I would say that one very important aspect that they did not apply is any kind of data-driven transplant, meaning they didn't say, oh, this non-responder would benefit most from this responder. They don't have those data. We don't have those data. So you would have to do a very large trial where eventually you can actually say, okay, we have a non-responder to this immunotherapy and we have 100 donor stool samples in the freezer. You can say computationally, this one donor is going to make the biggest difference in this acceptor, and you're going to take that poop and give it to the acceptor. That will, of course, require a lot of studies, a lot of data, but this is going back to that Manhattan Project, right? If we all dedicated resources to understand that one question, of course we would nail it, because it's just biochemistry we don't understand. There is no magic. There is no bad luck. There is nothing weird. It's just science. It's just molecules interacting with each other, and we just need to understand which are the molecules that are actually doing this. Now, if we were to, let's say, mount a Manhattan Project against any kind of cancer, I would want to focus on what are the physiological pathways that lead to melanoma, so that we can prevent it, instead of let's spend 5 years treating cancer, right? That would be a more noble cause to actually go after prevention, because if you can prevent cancer like we can prevent the Black Plague, you don't need to figure out how to treat the Black Plague. You just prevent it, right? I don't wake up thinking, oh my God, am I going to get Black Plague today, right?
Yeah, and thank goodness for that. How does it tie into, your academic work and the spin out that you had into Viome, what tools have you developed, the platform, and could you walk the listeners through some of the key learnings through your research?
Yeah, the key learnings were I was the chief scientist at a large genomics facility. So we spoke earlier about the Human Genome Project. Los Alamos National Lab was one of the centers where the Human Genome Project took place. So I was in that genome center, many years after the Genome Project finished, but it was still a very large genome center, and I was a chief scientist there, and several things blended together at the same time. First, I had this devastating autoimmune disease that was going to send me to a wheelchair. And if you know me, you know that I am a ball of energy. I have hiked thousands of mountains. I am like hyperactive. I am just literally a ball of energy. And to me, the thought of going to a wheelchair was simply unacceptable. And I didn't accept bad luck, right? So many people have autoimmune disease and they're like, oh, bad luck. Now I'm going to suffer the rest of my life. Well, that wasn't me. So my ability to identify the root cause of this autoimmune disease, and that I was able to change my diet and completely cure myself, and I mean completely, was one big factor in this sort of awakening that I had. The second big factor was this understanding of what academia are actually doing and what pharma is actually doing. And we discussed that earlier, that academia are not solving the problem. Pharma is not solving the problem to a large extent, right? They're just doing their own thing that they're incentivized by. So that was a big realization. And another realization was that, hey, I can solve this problem. I have the right background. I have the right passion, the right motivation. I can solve this problem. So in 2010, I basically quit all of my scientific projects that I've been working on. I basically reset my scientific career, which every single person around me said, that's a scientific suicide, okay? They said, in 2 years, you will be a manager and you will have quit science because this is a scientific suicide. And I said, watch me. And so in 2010, I completely reset my career. And at that time, what I had already known was that the human physiology is all about functions. It's not about who is there, which means it's not which genes we have. We all have the same genes. You and I have the exact same genes, but how my genes respond to some inputs may be different from how your genes respond to some input. And my genes are going to respond to two different inputs very differently. And we need to understand the activation of the human genes. So I knew that I needed to focus on the functions of the genes and not the composition. That's one big thing. The second big thing, I knew that microbiome is going to play a big role and nutrition is going to play a big role. And it's really this flood of nutrients that comes in that your physiology and microbial physiology process to build your health and build your disease, right? And so if you just think of yourself, you're 3D printed based on nutritional ingredients, using activation of specific genes, and how you're 3D built, you can be 3D built into a healthy human or a diseased human. It's really that process of converting nutrients into your building blocks that's going to determine your health and disease status. And so we needed to understand how nutrients are processed into those building blocks, and that leads to health and disease. And so in 2010, I also ran a genomics lab where we were profiling the microbiome using the standard method, which was 16S sequencing. And at that time in my lab, I already knew its weaknesses. I already knew that we could tune we could tune the results output simply by bioinformatic knobs, right? You can look at the same data and adjust the bioinformatic knobs, and you can get completely different results. And we can dive into that deeper, but it's really sort of a very nuanced technical approach. But I knew that 16S profiling was not important. I knew that metagenomic profiling was not important, and now we know very well that that's the case. Metagenomics tells you who's there, but you have no idea what they're doing. And so, if your T cell requires butyrate in order to convert itself to a memory T cell, if your T cell does not understand the concept of Faecalibacterium prausnitzii. Your T cell doesn't have a brain, doesn't speak a language, doesn't understand that, hey, Momo, you did a gut microbiome test and your profile says that you have Faecalibacterium prausnitzii. I should transform into a memory T cell because Faecalibacterium prausnitzii is a good butyrate producer. My T cells have no ability to have that logic. My T cells are waiting for a chemical signal, a very specific molecule called butyrate. If I have a ton of Faecalibacterium prausnitzii, but it's not making butyrate, I am not going to get a memory T cell. No matter what the taxonomic classification says, no matter what my stool test says. So we have to understand how much butyrate is made, not the fact that I have butyrate producers that could be producing butyrate, but I have no idea if they actually are. And so that was really a fundamental thing. And so if you look at the functional profiling methods, they are metatranscriptomics, which is RNA sequencing, metaproteomics, which is protein sequencing or protein analysis, and metabolomics, which is understanding of these metabolites. And in 2010, as is the case in 2024, 14 years later, metaproteomics and metabolomics are simply not technologies that are user-friendly, that are cost-effective in order to profile, digitize the human body. That's one massive problem.
Sorry, could we just unpack that just a little bit? Could you just explain to the listener why they're not well suited to kind of wide-scale rollout in terms of cost, complexity?
It's a highly technical discussion, but basically the sequencing technologies for proteins have just simply not been developed like next-gen sequencing has been for RNA sequencing and DNA sequencing. So that's basically the bottom line. The technology has not been developed for high-throughput global provision of tests to a consumer who self-pays for the test, right? So you can do metaproteomics in a lab. Like if you had a Manhattan project, you're sure as hell would be doing metaproteomics. But that is a lot of money that individuals cannot pay for. Same thing with metabolomics. It's a very costly, very highly involved. We're processing now tens of thousands of samples per month in a fully automated metatranscriptomic lab. If we were doing metabolomics, it would cost 10 times that amount and be slower, and no one's going to pay for that. That's one technological drawback. The physiological drawback of metaproteomics and metabolomics is that while you can measure those metabolites and proteins, you don't necessarily know what caused their production. And so what I really like about metatranscriptomics is that it connects the composition with the function. It gives you both. And so when we sequence RNA from the gut microbiome, we quantify the RNAs. We know what the functions of those RNAs are, that they're producing butyrate or whatever other metabolite, but we also know which microorganism they came from, so that if we want to tune the composition of the microbiome via probiotics, for example, we can do that. And so in 2010, the state-of-the-art metatranscriptomic technology was such that it was not affordable to even scientists. And 14 years later, it's still the same. The vast majority of scientists do not have access to metatranscriptomics because it's still so expensive and so complicated. And so in 2010, I said, we have to develop metatranscriptomics that's going to become fully automated, scalable, clinically validated, and affordable. It has to be affordable because otherwise it doesn't work. So today we have 600,000 customers in 100 countries because it is affordable, because it is scalable. And so from 2010 until 2016, I raised about $50 million in grants from mostly the US government and had a team of about 35 to 50 scientists that worked hard on this problem. And again, I was at that time very much driven to develop the technology that will spin out into a business and be what Viome is today. I was not driven by publications at that time. We did publish, but that was not the primary objective. And so every single compromise that we had to make in the lab had to do with, I want this to be cheap, scalable, and globally scalable. And the challenges at that time were such that everyone told me, this is not possible. Like, you cannot do that. Like, RNA is so unstable. You cannot collect RNA in the UK and ship it to the US and maintain its integrity. That's just not possible. And they said, you can't have an automated lab where RNA is like exposed, for minutes because it'll degrade. And you can't do this and you can't do that. Everyone found these fundamental problems. And I just said, don't worry about it. I'll take care of it. Now, it took 6 years to develop this technology, and the amount of work that went into those 6 years, I can write a 600-page book that summarizes the amount of work, the amount of hustle, the amount of painful compromises, the amount of barriers we had to overcome. But in 2016, we had the key pieces, which were we had the ability to collect stool samples, saliva samples, and blood samples anywhere in the world, ship them anywhere in the world where the RNA is preserved, and very importantly, all pathogens are inactivated. And that's very important, because if you're shipping blood and you cannot provide data that pathogens are inactivated, that's going to take a major hurdle, financial and time, to get to the lab. And then we had sample to result, all processes that make very efficient, high quality metatranscriptomic analysis for very low cost. And so in 2016, my team had to basically overcome all technological challenges. There was one thing that the vast majority of people don't know, would never think to ask, and so on. But there is a part of our technology that we solved that's called barcode hopping. I don't know if you know about index hopping or barcode hopping with next-gen sequencing on Illumina platforms, but basically, it was a huge thorn in my side. If you take one sample, and that one sample, let's say blood sample, I mean, this was actually the case. I worked in Kenya at that time. I had a project in Kenya. So we brought a blood sample from a child who had malaria, and we sequenced it together with 25 blood samples from people in New Mexico. And the bioinformatics came back and said all 26 people had Plasmodium falciparum in their blood. And I said, well, that's not possible. Like, I know that these 25 people from New Mexico do not have malaria. That's a fact. And so we dug deeper and we identified this problem called index hopping, where when you sequence multiple samples on Illumina at the same time, there's about 1% crosstalk between the samples, which means that every sample that you sequence, 1% of that data did not come from that sample. So you have 1% noise. The problem is that the vast majority of molecules you're measuring, their relative abundance does not reach 1%. It's well below 1%. So you have 1% noise, and your signals are in 0.01, 0.001, 0.0001%. And for machine learning, that's not going to work. You can't basically, those data are not suitable for machine learning, because the machines are going to pick out the noise and not the signal. And so we worked very hard for 3 years. This is now patented technology, where we moved that noise floor by about 10,000-fold. And now the vast majority of the molecules we measure are actually above the noise. And that's really one of many aspects of this technology that took 6 years to work out, to where I was comfortable leaving my really, really cushy government scientist job and spin out a company that had a 95% chance of failure, right? Like all other companies.
You've been on the roller coaster ride since 2016 building the business.
Yes.
And that's probably a whole separate conversation.
Yeah, that's a huge conversation, yes.
Yeah, and well done to you, because it's super hard. What I'd like to pull out from your brain and your expertise is what are some of the things you've learned now, having applied metatranscriptomics to so many different samples, and how are you building your evidence base and thesis within the company about how do we get to preventing all disease?
Yeah, I mean, we can discuss that question for several hours. But we do not have several hours. So let's sort of pull out some takeaways. So one takeaway is that we are an AI company. So today, when a customer submits their stool, blood, and saliva samples, they undergo what I call biochemistry and math. There are no humans involved. Until the customer receives their results and recommendations, there is no human involved. It is 100% objective science. So the biochemistry refers to the metatranscriptomic lab analysis, where we currently generate about 22,000 data points. So everyone goes to the doctor and measures their, let's say, metabolic profile, and you get your lipid panel, and you get your fasting glucose, fasting insulin, maybe CRP, and a few things. So you go to your doctor and they draw blood, and you get, let's say, 7 or 8 data points. Our current test generates a little over 22,000 quantitative data points. And so that's the biochemistry, but that number keeps increasing every year, and it'll end up in the hundreds of thousands, right? Because that's how complex the human body. You cannot profile the human body with 4 data points. That's not going to work. So we generate more than 22,000 data points, quantitative data points that are relevant to human health, and then mathematical algorithms take over and translate those 22,000 data points into results and recommendations for every customer. So that's the process. Now, one big takeaway that I would like everyone to understand is whenever you hear someone say, we use AI, okay, the most important question that you need to ask is, what data is AI using to generate the output? How was AI trained on what data set? What were the metadata? What were the data? Were they produced using clinically validated tests, or were they using some researchy test? Were they just using publicly available data that you found online? And those are the most important questions about AI, not about what computer you run the AI on and what are the algorithms. How you trained AI is the most important thing, because if you trained it on wrong things, even though it's fancy AI, it will be giving you wrong answers every single time. And so we understood that from the beginning. And so I'm also the head of clinical research at Viome. In the last 7 years, we have enrolled more than 16,000 clinical research participants in our clinical programs. Wow. Now, that's an extraordinary effort. And we have mined the data from 600,000 customers. So we have a petascale level data lake that our machines are using, where every person, when they collect any kind of samples for Viome, they also answer questionnaire, which includes not only what medications they're on, what diseases they suffer from, what symptoms they have, but they also complete 36 clinically validated surveys that actually quantify any disease that they may have at the moment at which they're collecting their samples. So when you have someone who's been diagnosed with IBD, there's a dramatic difference whether someone is undergoing an IBD flare or they've been in remission for 10 years, a dramatic difference. And we get those metadata. So we get very accurate molecular data, clinically validated, and we get very accurate metadata, also clinically validated, all going to one data lake that's used for training of our algorithms. So that's one extraordinarily important thing that no other company that I'm aware of in this space does anywhere near what we do, because that is what AI runs on. It runs on solid data. It doesn't run on air or, let's just scrounge the internet and, find some stuff.
What you have is not available anywhere else. It's been hard earned. And it's, in part you're offering a great service, and it's also from all the patients who have slightly different profiles. So I guess over time it's going to get better, right?
It is getting better. So we talked about TMAO, right? We talked earlier about the example of converting choline into trimethylamine, right? Those pathways that did that conversion were not known 4 years ago. So our computers were not aware of that. We couldn't give our customers a TMAO score because we simply didn't know the pathways. But now we know. 2 years ago they were published and we incorporated into them. So every month there are new learnings, both internally from our own data and from external data. Whenever a microbial pathway is found for something, we're like, oh wow, that's great. Let's plug it into our platform. So yes, we are still on the exponential growth in terms of learning about the human body and the microbiome. That's how much more there is to discover, right? So it is one of the major aspects of our platform is that it's continuously learning. Every new customer benefits from all previous customers. It's not like your blood test that you go to the doctor and you do the lipid panel and you do not benefit from the previous customers because the lab that performed the test, they don't have any science, they don't have any data lake, they don't have any AI. They're just cranking out the data and handing them to you, and that's it. And so that's not what we want. Our platform is all self-learning and continuously learning.
And is it your vision that you would be able to prevent all disease, almost all disease, through diet alone? Is it possible to do that if you start very early in life?
Exactly. The key is almost all diseases. If someone has a genetic defect where a protein has a mutation and it's not as functional as a normal protein, that's going to have to be addressed with CRISPR or some kind of a genetic modification, because you simply have a genetic fault and there is no, maybe there's a genetic fault where you can overcome it with food because it's a minor fault, but something like, rare genetic diseases, you will have to fix that at the genetic level. And why not? We're now developing those technologies to enable you to do that. But regardless of the genetic content, healthy genetic content, meaning you don't have a genetic issue, yes, you will be able to heavily modulate your human body. And the reason I say that we will be able to do that for these non-genetic diseases is that nature has solved this problem. It's not like we're inventing something new. We're literally just figuring out how nature works, because there are tens of thousands of examples today of people in Japan and Greece and Corsica that are 100 years old, and they literally have no mental, physical, or cognitive symptoms. They function normally like a 40-year-old in a developed country. So nature has already figured this out. They don't have IBD, they don't have depression, they don't have cancer, they don't take any pharmaceuticals. So nature has already figured out how to live in harmony with your environment. All we have to do, and that's what we're doing at Viome, we're decoding nature and then empowering every single individual to have that life that a Greek person has, and that's it. So we're not really, there's no science fiction. This is reality.
I'm not sure if you've spoken about it before publicly, and you may not be comfortable talking about it, but there are patients who listen to this podcast. Would you be comfortable sharing any more about your own experience and how you actually managed to cure it?
I mean, this is an interesting story. I'll give a summary, and then if you think you're curious about something, we can dive deeper. But basically, the mammalians, so mammals, right? Mammals are a type of organism that live on Earth that is very diverse today, but we came up, let's say 70 or 80 million years ago or so, just prior to the extinction of the dinosaurs. So all mammals had the ability to produce two types of sialic acid. And sialic acids are these carbohydrates that are produced by our own genes, and they are secreted to the outside of the cell. They basically make a cell wall on the outside of human cells. It's called extracellular matrix, or ECM. And these sialic acids have a function in cell-to-cell communication and forming sort of these layers. But it turns out that sialic acids are great handles for pathogens to enter our cell. So if you look at many pathogens, they are using these sialic acids to gain entry into our cell. So 2 million years ago, all mammals had the ability to produce these two types of sialic acid. But humans, Homo sapiens, well, there was no Homo sapiens 2 million years ago, but there was a Homo something, whatever it was at that time. There was some kind of an event where some pathogen killed off all humans that were able to produce this sialic acid called nu5GC. 100% of them went away. But there were humans with a mutation that inactivated the gene to produce this sialic acid, and those humans survived that event. And today, 100% of humans that live on Earth cannot produce that sialic acid, but all other mammals can. So cows can, and pigs can, and lambs can, and all of those mammals, right? And so I was born, just like you were, without the ability to produce this sialic acid called nu5GC. NEU5GC. However, that molecule is found in all mammalian foods. So your beef, your lamb, your pork, your milk, and so on. And so my immune system, mine, maybe not yours, but my immune system did not learn to tolerate this molecule. And that's possible because we were vegans when I grew up. When I was raised, when my immune system was developing, I didn't have exposure to animal products. And so my immune system did not learn that that's self. And so when I started introducing it, then it said, oh, this is foreign, and it reacts to it. So basically, when I consume any mammalian food product, nu5GC is transported to my cells, and my cells have all the other genes for incorporating nu5GC into the extracellular matrix. I can't make it, but I can transport it and integrate it into the extracellular matrix. But my immune system says, wait a minute, this is not self, and it reacts to it. Now, this nu5GC is integrated into epithelial cells, and that's in the joints and in the arteries and some soft tissues. And so basically, if I eat mammalian food products, my immune system attacks basically my entire body. And that over time, it degrades it. And so, this started in grad school, and fortunately, this sialic acid is not very immunogenic. So it causes a very mild immune response. It's not like a pathogen that just completely invigorates, but it's like a slow, chronic inflammation that just chips away and eats away at your body. And so I started developing symptoms, even though I had probably had the same thing for many years, but I started developing symptoms in grad school. And then 10 years later, basically my joints were just feeling like I was 85 years old. They were clicking, they were grinding. My right hip was locking. I had to cancel many hikes because my right hip would literally just lock up and it wouldn't move. And so I stopped eating. So, I realized this mechanism from a bunch of publications. I stopped eating mammalian food products. And because the half-life of this new 5GC in extracellular matrix is very long, it's about a month. It took about 2 months to be convinced that this was it, but I had to be 100% clean. I mean, 100% clean. But I understood the biochemistry. I understood that I needed to be 100% clean. I stopped eating mammalian food products, and in 2 months, I was like, this is it. It took just over a year for all my joints to heal. But since then, no disease whatsoever. I did test myself on 3 separate occasions, 2 intentionally and 1 unintentionally. I consumed mammalian products to see what happens, and all the symptoms come up in 48 to 72 hours, and they get worse over the next 2 weeks, and then they start declining, and after a month, they clear. It is an amazing thing.
Wow, you've self-diagnosed yourself and fixed it, which is commendable and amazing, and it ties into the mission and the vision for Viome as well. And one thing we didn't touch on, though, was some of the recommendations that come out of the analysis that the people that work at Viome are able to produce for the customers. So my understanding is it can help take a data-driven approach to your diet. Are you able to sort of go a bit further on that in terms of personalised dietary recommendations and where we're at today in our knowledge base and where we'll be in the future?
Yeah. So the AI is all pre-programmed and learned what molecules are. So the major outcome of all of our clinical research, whether it's on the 16,000 clinical research participants or on the 600,000 customers, they're all in one data lake. We train machine learned models to understand what are the functional activities or the biochemical activities of the microbiome that are associated with health, and what are the biochemical activities that are associated with disease. And we also leverage the knowledge in the literature because, for example, we know TMA is bad for your health. We don't need to run a clinical study to do that. That's a well-established fact, right? We know that oxalates that are in plant foods are bad or can be bad for you. We don't need to establish that fact. That's already been done. So we're leveraging all the domain knowledge as much. And so we train our computer to say, oxalates are bad for you. And so that I'm going to give you the most recent example of my test. So we measure the processing of oxalates by each person's microbiome, and we get an oxalate score. We output an oxalate score to every customer. And so for years, I've been testing, and my oxalate score has been good. I'm mostly vegan, so I depend on soy a lot. I used to eat 3 meals with soy. But about 2 months ago, the score went below optimal, so it went to suboptimal. It's not bad, but it's suboptimal. And what the computer automatically said is it automatically recomputed my diet and said, okay, soybeans and black beans are high in oxalates, and so is spinach. Spinach is very high in oxalates, so it puts spinach into avoid list, so do not eat any spinach. Soybeans and black beans, right? Because of, for me, just for me, soybeans and black beans contain quite a bit of oxalate, but not as much as spinach, so minimize them, meaning eat some, but not eat a lot. But lentils and chickpeas don't have much oxalate, so go ahead and move them up to enjoy and superfoods. So that's what the computer decided for me. So it basically shifted me from eat anything you want with oxalates because my gut bacteria were degrading them very effectively to remove spinach from your diet, minimize your soybeans and black beans, maximize your lentils and chickpeas. And so that's a data-driven decision that no nutritionist can tell you, no doctor can tell you, unless you measure the molecular physiology of your microbiome. There is no other way to know what is going on at the molecular level in your colon unless you do this. And so what that did is, well, at first I was disappointed. Not as disappointed as people who are told by the computer not to drink coffee, but pretty disappointed because
goodness me, I couldn't do that.
Basically,
I'd have a problem.
See, that's a serious problem if you cannot have coffee. But luckily, that's not for me.
Big problem.
But I take this stuff seriously because I know that this is real, and I know that this is measuring our human physiology. So I took this seriously, and I went to my kitchen and experimented for a weekend, and actually came up with recipes now to make tofu from lentils and chickpeas. That it turns out now in retrospect, I should have done earlier, because this tofu that we make at home, yes, you cannot buy it, you have to make it, but it's not too much of trouble to make it. But it's whole lentil and whole chickpea tofu, which means it integrates all the protein and all the fiber from those legumes, whereas soybean tofu does not, in fact, maintain all of the fiber and all the protein from the original soybeans. So it's a processed food. So these are whole foods, which is better, and it actually tastes better. It has more flavor. Tofu is basically tasteless, whereas if you just bake the lentil or chickpea tofu, you actually can enjoy it on its own. It tastes very good on its own, but it tastes even better in different meals that we make. And so I experimented for a weekend with different ratios of different things, and boom, we came up with this recipe, and all of my family says these are better. So now every weekend I make 2 to 3 batches of lentil and chickpea tofu, and then that's what we use during the week for lunches and dinners. So now I only eat soybean products for breakfast. I make soybean yogurt for myself. It's a vegan yogurt that I enjoy, and I eat that with my granola every breakfast. But then for lunch and dinner, I no longer eat soybeans. I just eat lentils and chickpeas.
So it sounds like you do a lot of your own cooking and you make stuff from scratch. Do you have thoughts on big food?
Go ahead.
You have thoughts on the big food industry and how they're sort of big food industry and ultra processed side of things? Could we just talk about that a little bit, and then we can move to some forward looking thoughts in the future?
Yeah, I mean, the food industry's bottom line is how much money they make, first of all. But second of all, they have to compete against others, right? So if they make a product that's good for you, but it doesn't sell, well, that company goes under. And so they unfortunately have to compete in the marketplace where people want cheap food, right? And that's been the main driver over the last few decades, maybe possibly even 100 years. The driver of food was cheap food because no one understood the importance of how important healthy food is for you, that you're basically 3D printing your body out of the food ingredients. We didn't know that. And so now that we know that, I think there is a shift towards, people are actually now selecting food that is more expensive, but that is better for them. So whole food plant-based diets, and meats that have no antibiotics, and foods that have no preservatives, and so on. So I think that the food industry will adjust to the new needs, and it's a transition period. The food industry will evolve because they're there to respond to the demand. And the demand has been, I want the most food for the least money. That's been the demand for the 100 years. And we got what we asked for, cheap food that was ridden with chemicals and not good for you, ultra processed. So that's a whole other topic that we can have in the future on, how to go shopping, how to cook, what to avoid. We can have a whole new podcast on just focusing on that, because it's a very important topic.
It's really, really hard to know what to do. There's also the microplastics discussion to have as well, and how you can try and avoid that, because that's definitely a problem.
Yep.
And there's lots of conflicting information. I personally, when I'm speaking to people, and I really love this 3D printing concept. You're 3D printing your body through what's going into your mouth. I just think that's amazing. I've noted it and I'm going to use it. But very often when I'm speaking to people, because it's similar to you, I do a lot of making stuff from scratch, shop organic, try and avoid ultra-processed foods at all costs. Although some ultra-processed foods are not as bad as some others based on the definition. Like Greek yogurt, for example, is actually a processed food, but it's not within that bucket of the ones that are micronutrient light and contain, So, if I could bring it back to the blue zone discussion we were having very briefly, how important is the food aspect, do you think, in living to 100? And how important is the exercise, low stress, sleep, Could you attribute a level of importance, or are they all just absolutely fundamental?
I mean, if you get 4 hours of sleep a night, I don't care what you eat, I don't care how much you exercise, you're not going to live a long and healthy life. So yeah, I think that food, exercise, and sleep and low stress environment are all so important that I really would rather not prioritize them, because I don't want anyone to say, well, I'm going to sacrifice on stress or sleep or exercise, because they shouldn't. No one should sacrifice any of those four. They're the pillars of our health. And if you have a chair with 3 legs, it's not going to sit. You have to have all 4 pillars. And that is food, exercise, sleep, and mental health. Being in the right environment with the right positive people, surrounded by love, care, support, all that. That is, I can't imagine functioning without that, or without enough sleep, or without exercise.
These are the fundamentals.
4 pillars of health, man.
So, yeah, and the last episode that I released, we spoke about these fundamentals, and this person was a kind of biohacker, wellness expert, and we did talk about some of the fancier stuff, but what it all came down to was what we described as the fundamentals: the sleep, the exercise, the relationships that you have with people, and what you put into your body. It's an investment. If you take the time to prepare something that's full of the right micronutrients and the fiber, and that doesn't contain harmful emulsifiers and things like that, you're doing your body a good service, and you're setting yourself up for success. And I guess that's probably a good note to wrap up on. This has been a super interesting conversation. We've unpacked a lot of key themes. I've got some analogies to take away. I know the listeners will have loved it. You're actually probably the most requested guest so far in the podcast from listeners. So you've obviously got a profile within the microbiome space. So thanks for agreeing to do it. Thank you for your time. Thanks for imparting your wisdom. I look forward to hiking with you at some point in the future. That'll be cool.
Yeah.
And thank you for supporting the podcast and the mission, and for all your great work.
Thank you, James, for inviting me, and thank you for driving the field forward. We need more information flow and more understanding, and make the world a better place one day at a time.